Early, portable detection of tumor metabolic markers enables faster triage and better outcomes. We target DLA plus PSA to raise specificity for prostate cancer risk. Smartphone readout allows bedside electrochemical and colorimetric interpretation. Feature 1, biomarker thresholds, DLA 4 to 12 millimolar, PSA over 4 nanograms per milliliter. Feature 2, dual detection, electrochemical plus colorimetric. Feature 3, screen-printed electrodes and low-cost strips for point-of-care. Feature 4, clinical correlation, DLA versus Gleason score, RO equals 0.78. Designed for clinical researchers and lab technicians in hospital and decentralized settings. Use cases include outpatient triage, population screening pilots, and intraoperative margin assessment. Next, step-by-step protocol, hardware schematic, and cross-platform app integration. We will cover sample prep, strip design, detection chemistry, and software logic. Begin with the reagents, enzyme chemistry, and calibration steps. Step 1, reaction setup, reagents and concentrations. First, sample 50 microliters, add DLDH10 UML and NAD plus at 1 millimolar. Second, add color reagent, NBT 0.2 mg/ml with PMS 0.1 mm. Incubate 15-30 minutes at 37 degrees Celsius. Third, for paper strips, pre-immobilize enzyme and dye. Dip for 5-10 minutes. Color mapping, 0.5 mm pale yellow, 5-15 mm light purple, 15-30 mm dark purple. Use smartphone RGB analysis, for example GB ratio, to estimate DLA concentration. On-device model runs via lighter T for low latency inference and on-device privacy. If DLA 6 and PSA 4.0, status =sign prostate cancer. High risk. Example flutter function. Analyze prostate reads PSA from electrochemical sensor and image-derived DLA. Convert and quantize the model with the lighter T toolchain for sub-megabyte runtime. Electrochemical sensors detect NADH oxidation currents corresponding to DLA enzymatic turnover. Screen-printed electrodes, low cost, reproducible, compatible with disposable strips. Calibrate across 0 to 30 millimolar with LC-MS/MS reference for accuracy. Use LC-MS/MS as ground truth to establish D/L lactate separation and correct bias. Check cross-reactivity versus L-lactate, pyruvate, and common interference. Troubleshoot, increase NAD plus concentration for weak signal. Add L-lactate control to detect bacterial confounders. Final hardware flow, sample the sensors, microcontroller digitizes signals, smartphone performs inference and displays risk. Results can be stored locally or exported to EMR systems, preserving PHI on device when required. Validation, correlate DLA values with Gleason scores. Observed RO approximately 0.78 indicating strong association. Method comparison, colorimetric strips for low resource screening, electrochemical for sensitivity, LC-MS for definitive analysis. Tissue-level DLA ranges differ. Colorectal and pancreatic often show highest tissue levels. Serum levels are 100-fold lower. Clinical threshold example, DLA, 6 mm and PSA, 4 ng/ml signal high risk for prostate follow-up. Server-side or research logic, if D_lay_mm greater than 6 and PSA_level greater than 4, label prostatic high risk. Deployment checklist 1. Calibrate against LC-MS 2, validate cross-reactivity 3, manufacture strips 4, certify clinical study 5, integrate with EMR. With careful calibration and validation, smartphone DLA diagnostics can extend metabolic cancer screening to new settings. References and implementation code snippets available in the video description. Contact the team for collaborator access.